All right, so welcome to part 2. So again, we can characterize operons in two ways, whether they are normally off or whether they're normally on. If they're normally off, we call them inducible operons. If they're normally on, we call them repressible operons. The other way to characterize operons is what the regulatory protein is, really by what it does. So if the regulator is an activator, which means that it turns the operon on, we call this positive regulation. And if it's a repressor, that means that it turns the operon off, and we call this negative regulation. So we have 4 possible ways of characterizing operons. Let me move this up a little bit. So 4 possible types of operons. We can basically have positive inducible. We can have negative inducible. We can have positive repressible. And we can have negative repressible. So those are 4 ways that we can characterize operons. Okay, so let's think about this. If our regulator, which sits on our operator, typically, is an activator, that means that it's turning the operon on. And we have positive regulation. Now, whether it's inducible or repressible depends on whether or not that regulator, the activator, is sitting there normally or if it's not. So let's just say we have an operon that is generally off. What kind is it? Is it, let me go back. Is it inducible or is it repressible? If it's normally off, it's inducible, which means that we have to turn it on. If the regulator is an activator and it's normally off, that means that the activator is normally not on the operator. It's normally not on the operator, which means that our activator is inactive. So here, this would be positive inducible, which means we have inactive activator. That needs to come in and once it becomes active, then it can sit in the operator and turn on the operon. We're going to talk about this with actual illustrations, but I just sort of want to go over these concepts. This is why I was hoping that it might be a good thing that this video is actually a video rather than an actual lecture, because then you can go back and hear it again. This is not easy. There's a lot of stuff to keep in mind. Okay, so just as an example. Okay, so if we have negative inducible, that means we have a repressor, and if we're inducible, that we're normally off. And if we have a repressor on an operon and we're normally off, that means that the repressor is typically on the operator. Okay, so under negative inducible, that means we have an active, what we call an active because it's actually functioning, repressor. Okay. Under positive repressible, now positive we have an activator, but repressible means we're typically on. So if our operon is typically on, we're being regulated by an activator. That means that the activator is typically on the operator, keeping it on. All right, so that means we have an active activator. Now, under a negative repressible, there's only one other option that we haven't used yet. Under negative repressible, we have a repressor because we're negative, and repressible, which means that we're normally on. So if the operon is normally on and we're regulated by a repressor, which turns it off, that means that the repressor is not normally on the operator, which means that we have an inactive repressor here. Okay, so let's move on to actually seeing what's going on here a little bit more. So we have positive inducible, for example, here. Again, it is inducible, so the gene is normally off. Gene's operon is normally off. And here the regulator is an activator, which is why we know that it's positive. Positive inducible regulation. Okay, so in this case, here is our operator. Transcription is normally off because it's inducible. And we have, they're calling this the promoter because here's our little pink capsule polymerase, RNA polymerase. And normally our activator here can bind. So again, positive inducible, we have an inactive activator. In order to activate the activator, you need some sort of substrate. This is called our, oh my gosh, what's it called? This is called our inducer. So here is basically our little molecule which binds to the activator and allowing it to become active, and this guy's called an inducer. So positive inducible operons initially have an inactive activator. It is activated activated by the inducer. Okay, and then it can activate transcription. So let's see. Okay, so we'll move on for now. So well, all right. So I'll just say right now that inducers well, let's say, okay, so inducible, I'll write it up here. So inducible pathways, inducible operons, tend to be involved in degradative processes to mediate where things are being broken down. They can also be called anabolic processes. And this is because they're often, you know, for example, when you're digesting something, that is a degradative process. And you don't necessarily want to be having the enzymes necessary for that process on all the time unless the substrate is present. So inducible operons, they tend to mediate degradative processes, and they are induced when the substrate is present. So here, for example, we have our substrate now that needs digesting, and so we're going to use that to activate transcription of our operon. Okay. All right, so that's positive inducible. Let's talk about negative inducible. So again, because we're inducible, our operon is initially off, and because we're negative, we have a repressor. So again, if we're negative inducible, our repressor is normally actually on our operator, is normally attached to our operator, keeping transcription off. Now, again, we have an inducible operon, tends to be involved in degradative processes. So a substrate, when the substrate becomes available for degradation, then the substrate itself activates, well, rather, let's say, it almost kind of deactivates our repressor. It kind of takes it binds here, induces a conformational change that allows it to leave the operator. So here it's going, leaving the operator. And this is called a co-repressor. We can call it inactivated by our co-repressor. Okay, so that's negative inducible. Here we have positive repressible. Now positive means that we have an activator. Repressible means, repressible means that it's typically on. Okay, repressible means that it's able to repress, so it's typically on. And so our activator here is normally attached to the operator. Now, in these diagrams, the operator tends to be upstream of the promoter, whereas in the first operon diagram that I showed you, the operator was downstream of the promoter. It doesn't matter. They can be in either place. But typically, the operator is going to be right next to the promoter because it's physically blocking the polymerase from attaching to the promoter and initiating transcription. Okay, so normally these guys are on, and the activator's on there keeping it on, and then at some point it gets deactivated such that this guy comes along, binds to the activator, and induces a conformational change that allows it to leave, turning transcription off. Now, repressible, where are we? Repressible processes, okay, tend to be repressible operons tend to mediate biosynthetic pathways. So they're catabolic. And they are, they're induced, the change is induced. So basically, let's just say they're repressed when there is enough product, end product of the pathway. So here, under inducible operons, they're kind of co-regulated by the start of the pathway, by the substrate. In repressible operons, they're co-regulated by the end product of the pathway, and they turn it off. So, for example, here we have some imaginary pathway where we're turning the purple circle into the blue diamond. Now, when we've got enough of the blue diamond is going to be able to sit on the activator, induce it to leave the operator, and turn transcription off when there's enough of it present, okay? So these tend to be pathways that are synthesizing molecules. So that's positive repressible. And again, still it's, this is still a co-repressor. Because it's still repressing.